Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Digestive infections can wreak havoc on the enteric nervous system, leading to persistent digestive issues. But, if there is an upside to them, it's that they may steel the gut against future assaults. The intestinal neurons of mice previously infected with either intestinal worms or pathogenic bacteria were more resilient to Salmonella infections, researchers report November 11 in Cell. Unlike intestinal epithelial cells, whose snappy turnover helps the gut rapidly heal from tissue destruction, intestinal neurons usually suffer lasting damage during infections and inflammatory conditions such as irritable bowel syndrome, IBS, or Crohn's disease. Since these neurons are important for everything from controlling gut motility to hormone secretion, this damage has enduring and often painful consequences. This is a classic example of disease tolerance, Mucida explains. A pathway that doesn't control how many pathogens are in your body, but rather how the body deals with inflammation and the tissue damage caused by the pathogens. So the researchers returned to the Salmonella bacterial infection model they used in their earlier paper, but with a twist. This time, they infected mice with a different type of gut bacteria, waited 3 weeks for the infection to clear, and then infected the mice with Salmonella. The mice that had been pre-infected had less neuronal death, more protective macrophages, and improved gut motility compared with mice that were only infected with Salmonella, even though both groups of mice had the same amount of bacteria in their guts during the second infection, suggesting that the main advance that this paper brings is to show that there is a long-lasting disease tolerance effect induced by infection, says Mucida. And this memory-like phenomenon wasn't specific to bacterial infections. To their surprise, the researchers found that pre-infecting the mice with intestinal worms, a completely different pathogen, triggered almost identical neuronal protection by macrophages during subsequent Salmonella infection, even when the bacteria were introduced 6 months after the worms were cl. According to their manuscript, infections of the parasite they used, Strongyloides venezuelensis, last 12 days or less. Closing parenthesis. In fact, the team's experiment suggested eosinophils were helping coordinate the neuron-protecting macrophages they had observed in their earlier paper. When eosinophils were depleted, there were fewer protective macrophages. The researchers further showed that 2 key cytokines produced by the eosinophils, IL-4 and IL-13, were essential for orchestrating the neuroprotective macrophages, and that adding these cytokines during Salmonella infection was enough to prompt neuroprotection in the absence of pre-infections. The fact that infection can be cleared for months, and you still see that protective effect that this infection had, goes against the dogma of innate immune cells not having memory, says Ahrens, although he notes that dogma is already being turned on its head by work on trained innate immunity. Rather, the immune system is there to maintain homeostasis. A trip to the pet shop. Mucida says finding disease tolerance in laboratory mice made the team wonder whether the phenomenon also occurs outside the vivarium. When I analyzed these mice, I saw tons of worms, says Ahrens. Accordingly, these mice had higher levels of eosinophils and macrophages, and they lost fewer neurons than the lab mice upon Salmonella infection, suggesting that a lifetime of exposure to intestinal pathogens had left their guts fully prepared to deal with assaults from gut bugs. I thought it was really cool that they analyzed store-bought pet mice, says Radboud University Medical Center translational cardiovascular immunologist Sarun Beckering, who was not involved with the study. She says that for this type of study, it's a great addition to the specific pathogen-free mice most biomedical researchers use in their labs, as their non-sterile life more closely models the constant bombardment with potential pathogens that people experience in their daily lives. I would really be interested to see if this has some sort of therapeutic potential in humans, says Beckering. However, she cautions that people shouldn't rush out to get worm infections to preemptively protect their gut neurons from future bouts of food poisoning. She says she wonders if instead of infecting people with helminths, Exposure to certain parts of the worms would be enough to trigger the neuroprotective effect, almost like a disease tolerance-inducing vaccine. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.